everyone. Today we are going to talk about hypothetical sequences. So, what are hypothetical sequences? So, hypothetical is only a term which means that the data is not validated yet. So, a hypothetical gene means that the gene has been predicted by using computational biology tools, and a hypothetical protein means that the protein has come through computational translation, which means the protein has not been validated in wet lab by any of the techniques, and the gene has not been validated at the genomic level. So now the question is, how can you reach to these sequences? So in this video, we are going to cover how to reach the hypothetical computationally translated sequences. So the idea is very clear. Let's see how we execute them. So there is one place wherein we can go and find the hypothetical sequences, and one of them is UniProt database. UniProt database is also called as UniProt KB. UniProt KB stands for UniProt Knowledge Base, and this database in turn is divided into two. One is SwissProt and the other is Tremble. SwissProt is a database which has validated protein sequences. What do I mean by validated? Validated means that they have been seen in laboratory, have been validated by using wet lab technologies, and researchers have seen this protein in nature, while the Tremble database, which stands for Translated EMBL, is actually a total hypothetical database which contains protein sequences which have not been validated through any of the proteomic technique. These proteins in Tremble database have come through computational translation. So, in case you are planning to set up some research or you're planning to do something, some sort of computational validation with the hypothetical proteins, this is the place where you have to come. So, if I will show you both of them. Let me go to SwissProt first. If you will go to SwissProt, you will see these two sections. The golden star tells you, the golden entry tells you that the particular record is manually annotated, and which means that the protein here which we are looking at is actually validated in wet lab, while the blue entry means that the data is unreviewed and it is only computationally analyzed, and these records are still await full manual annotation. So there is a lot here which you can take up for your research. For example, you have many species here. If you are interested in some particular species, go to the particular species and look for the proteins which have not been yet validated. And once you understand the sequence, and once you understand the protocol, how you can do it in wet lab, you can set it. And otherwise, there are many, bioinformatics workflows which you can use to predict the function of the hypothetical proteins. So this section, I brought you to SwissProt, wherein you will see that entries are, they're reviewed, they're manually annotated. So now let's go back and let's go to Tremble. So these are automatically annotated entries and they have not been reviewed. So nobody knows whether the protein Tremble has is actually present in nature or not. So if I will click it, I will reach to this page, and you will see that all the entries are blue here, which means that these entries have not been reviewed. These entries have not been reviewed, and so to speak, nobody knows if it is correct or not. So you can pick any entry, and then you can start working on it. Set up the bioinformatics protocol on how you can, predict the function of the protein, and by using different type of computational algorithms and computational softwares and tools, you can finally have a, have a report on what you think the protein can be, and then then as a researcher you can set up the certain experiments keeping that protein family in mind and can validate it. So this is how it is done. So let me open it for you. So click any entry here. It says that its annotation score is 0 and it is the unreviewed entry, and this entry, since is hypothetical, so you're not going to see any of the data here because there's nothing known about this, right? So you can directly come to sequence, click sequence, and you'll come here. Otherwise, since there is nothing, so you will find the sequence early because these all headings are missing here. So if you will come here, you have to click FASTA and then you will reach to this sequence. Now you take up this sequence and put it up with your plan, and plan what you are going to do next for the prediction of the function of this particular protein. So I have a separate video on bioinformatics workflow for the prediction of the function of the protein, wherein I have discussed several softwares which can help you do that. So please go check that out, and if you will go down, you will see other similar proteins with the same names in the different species. So you know for sure that this protein is something, and it is present in other species also, and it cannot be a coincidence, right? So that means that one day if any one of them will be predicted, if validated, for example this one will be validated in acidobacteria bacterium, then the others will also be validated. So this is how it works. So this is the entry information, and the sequence was last modified in June 2021, which is this year. So folks, this is the place where you can come and look for the hypothetical sequences to start your research from. I hope that was informative for you. I'll see you again with the other videos. Thanks for watching.